Hello, I am Noelia Ferruz and I am a new Ramon y Cajal group leader at the Institute of Molecular Biology of Barcelona. My background, I am actually a chemist, but then I moved towards bioinformatics and I did my PhD in actually a kind of a different field of what I do now. I was working on high throughput molecular dynamics to try to characterize ligand binding events. But then I moved in my postdoc to the field of protein design. I went to Germany, to the University of Bayreuth, under the supervision of Professor Birte Höcker. And then in there, we worked in the field of protein design. So in particular, we were working on an evolutionary inspired method for protein design. Given that we have so many different proteins in nature and they exhibit such different topologies, one of the questions that has been posed is how nature came up with so many different functions and topologies. And it's been shown that actually in nature and evolution actually work by recombining and replicating smaller fragments and combining them into new proteins in a kind of combinatorial fashion. So based on that, the thought was that maybe we could also take those fragments and recombine them in new ways, kind of from new protein chimeras. And this work, before I arrived to the lab, this had worked very well in the lab. So the idea when I arrived was to, what about we try to find all these different conserved fragments in the protein space and see how they are and how many are there. So this is what I did, and then so I found all these very conserved fragments that evolution had been used to create new proteins, and then we put that into the fossil database. And then to allow for the automatic combination of fragments, I created the software ProdLego to kind of take these fragments and allow for combining them into new chimeras in a fast fashion and then rank them based on different metrics. After that, I actually kind of got very excited by all the recent advances in artificial intelligence. So 3 years ago, there were, well, now ChatGPT, for example, is a very famous example of a generative language model. But 3 years ago, the first GPT model came out. So I was very excited to see how well it actually generates English text in such a long, coherent manner. So I thought, well, what about if this is generating English text so well, what about we take this and try to generate new proteins? So I did that with a model that I called ProGPT-2, and then later on with another model that specifically designs enzymes. And the first results, our results in the lab were like show really good results. Like these models seem to be generating very fit sequences that express well, and they are globular. And besides that, they seem to have, they are an idealized compact structure, so they seem to have the possibility to accommodate for function. We're also currently testing enzymes, so we hope to have results on activity very soon. To establish my new group, I was hoping to find an institute with a clear expertise in structural biology, but in particular in the wet lab. Because I come more from a computational background, I wanted to be embedded into like an institute with the facilities and also the expertise to help me test the predictions that the models produced in the lab. In the next years, I plan to look in deeper detail at how these language models, but also other type of models like diffusion models or reinforcement learning models, how well they perform in the lab, and then go specifically for more biomedical and environmental applications. And then also, I think it will be really important to have a look at explainability, because these models are kind of like black boxes, but they come up with really good predictions and generations. So it will be ideal to see why they are performing so well, and then maybe let us learn something. So get the models to teach us what are those rational principles for design.